Continuation of the previous sessions about some immune cells like eosinophils. Eosinophils, like neutrophils, are motile phagocytic cells that can migrate from blood into the tissue spaces. Their phagocytic role is significantly less important than that of neutrophils, and it is thought that they play a role in the defense against parasitic organisms by secreting the contents of eosinophilic granules, which may damage the parasitic membrane. Then basophils. Basophils are non-phagocytic granulocytes that arise by hematopoiesis and function by releasing pharmacologically active substances from their cytoplasmic granules. These substances play a major role in certain allergic responses. Then mast cells. Mast cell precursors, which are formed in the bone marrow by hematopoiesis, are released into the blood as undifferentiated cells. They do not differentiate until they leave the blood and enter the tissue. Mast cells can be found in a wide variety of tissues, including the skin, connective tissue of various organs, and mucosal epithelial tissue of the respiratory, genitourinary, and digestive tracts. Like circulating basophils, these cells have a large number of cytoplasmic granules that contain histamine and other pharmacologically active substances. Mast cells play an important role in the development of allergies. Then dendritic cells. Identified in 1868 during a close anatomical study of skin by Paul Langerhans, dendritic cells were the first cells of the immune system discovered. A dendritic cell acquired its name because it is covered with long membranous extensions that resemble the dendrites of nerve cells. There are many types of dendritic cell, and at least four major categories are recognized: Langerhans disease, interstitial disease, monocytic dendritic derived disease, and the plasmacytoid derived disease. Each arise from the hematopoietic stem cells via their different pathways and in different locations. Hematopoietic stem cell, common myeloid progenitor, immature Langerhans disease, epithelial tissue, immature interstitial disease, non-epithelial tissue or monocyte, plasmacytoid disease precursor, then maturation and migration lead to Langerhans disease, lymph node, then immature interstitial disease, non-epithelial tissue, interstitial disease, lymph node display. Macrophage monocyte derived disease, differentiation and maturation will lead to plasmacytoid derived disease. Langerhans disease are in epidermal layers of skin, and interstitial disease are present in the interstitial spaces of virtually all organs except the brain. As indicated by their name, monocyte derived disease arise from monocytes that have migrated from the bloodstream into tissues. From the tissues, they can move through lymph to lymph nodes or cross back into the bloodstream and use it as an avenue of transport to lymphoid tissue. So, plasmacytoid derived disease arise from plasmacytoid cells. They play roles in innate immune defense and as antigen presenting cells. Although there are important differences in the functions and phenotypes of different varieties of disease, they all share a display of class 1 and 2 MHC molecules and the B7 family of co-stimulatory molecules. CD80 and CD86 are present. Dendritic cells also have CD40, a molecule that can influence T cell behavior by interaction with the complementary ligand on T cells. So, dendritic cells are versatile, occurring in many forms and performing the distinct functions of antigen capture in one location and antigen presentation in another. Outside lymph nodes, immature forms of these cells monitor the body of signs of invasion by pathogens and capture intruding or foreign antigens. They then migrate to lymph nodes where they present the antigen to T cells and acting as sentinels in the periphery. Immature dendritic cells take on their cargo of antigen in the three ways. They engulf it by phagocytosis, internalize it by receptor mediated endocytosis, or imbibe it by pinocytosis. Indeed, immature dendritic cells pinocytose. Fluid volumes of 1000 to 1500 mu m square per hour, a volume that rivals that of the cell itself. Although the process of maturation, they shift from antigen capturing phenotype to one that supports presentation of antigen to T cells In making the transition, some attributes are lost and others are gained. Lost is the capacity for phagocytosis and large-scale pinocytosis, expression of class 2 MHC increase, which is necessary to present antigen to Th cells, and there is an increase in the production of co-stimulatory molecules that are essential for the activation of new T cells. On maturation, dendritic cells abandon residency in peripheral tissue, enter the blood or lymphatic circulation, and migrate to regions of the lymphoid organ where T cells reside and present antigen. Then follicular dendritic cell. Follicular dendritic cells do not arise in bone marrow and have completely different functions from those described for the dendritic cells discussed above. Follicular dendritic cells do not express class 2 MHC molecules and therefore do not function as antigen-presenting cells for Th cell activation. These dendritic cells were named for their exclusive location in organized structures of the lymph nodes called lymph follicles, which are rich in B cells. Although they do not express class 2 MHC molecules, follicular dendritic cells do express high levels of membrane receptors for antibody, which allows efficient binding of antigen-antibody complexes. The interaction of B cells with the follicular dendritic cells is an important step in maturation and diversification of B cells.